Hey everyone, my name is ROSHINI THAVAN. I'm doing Bachelor of Computer Science, majoring in Bioinformatics, and currently I'm in my fourth year, second semester. Today, I would like to present my final year project 2, which is entitled Classification of Breast Cancer Using Deep Learning based on integration of single omics variations data with supervision of Dr. Azura Binti Esama. First of all, I would like to explain the chapter 1, which is introduction. So this chapter is a brief explanation on introduction, problem background, problem statement, research aim and objectives, research scope and contribution, and also report organization. So cancer is one of a complex disease which is caused by malignant tumors where the cells in the body divide in an abnormal way. So single cell variations, DNA methylation, and chromosomal abbreviations are just some of the genomic and epigenomic alteration that drive cancer, a diverse disease. So integration of single omics variations data integration play a very important role in improving prognosis and prediction accuracy for clinical phenotypes. So deep learning is applied in this research, which allows the computational models with multiple processing layers to learn data representation with multiple levels. So next, I would like to explain the problem background. So here, DNA mutations can transform normal breast cells into cancerous one. So correctly identifying, analyzing, and classifying benign and malignant tumor is currently very challenging. Early diagnosis of this type of cancer increase the possibilities of a correct treatment. Otherwise, it may lead to spread and affect the whole breast or other parts of the body. Identifying the accurate type of cancer is playing an important role to select the suitable treatment approaches according to the type of cancer suffered by the patient. But examining prepared tissue sessions to determine the type of cancer, whether the tumor is malignant or benign, requires a lot of time. This study will address the integration of single omics variations data using a selected integration method, and the integrated single omics variations data will be used in the classification of breast cancer using 1D CNN. So the research aim is to implement the 1D CNN method to classify breast cancer based on the integrated single omics variations data and evaluate the performance of breast cancer model development in terms of accuracy based on the classification result. The next one is a research question. So the research question is what technique can be used to enhance the evaluation of breast cancer classification findings by using integration of single omics variation dataset. So the research objective is to study the breast cancer and deep learning method performance using the integrated single omics variations dataset, which is get from linked omics website. And then to integrate single omics data and classify breast cancer using the integrated single omics variation data. And then the third objective is to evaluate the performance of breast cancer model development using 1D CNN by acquiring the accuracy of the classification result. Next, I would like to explain the research scope. So, classify breast cancer that has a higher rate of death among females using 1D CNN and the integration of single omics variation. The data set is took from linked omics, and the Cancer Genome Atlas linked omics contain 32 TCGA cancer types and 10 clinical proteomics tumor analysis consortium cancers cohorts with 15,136 overall samples. So next, I would like to explain the literature review, chapter 2. So this chapter is an overview of the literature review, which consists of several existing activities in data categorization using deep learning methods. So this part is divided into several, which explain the breast cancer classification and the techniques we use to diagnosis of cancer. So the information that gathered from the previous research is used to simplify the research conducting process. So this chapter examines deep learning algorithm feature selection and integration method based on the previous research. So the proposed solution is in order to classify the subtype of breast cancer, integration of single omics variation data analysis is applied because it can use more information than single omic data analysis. It also allows to identify common molecular features across multiple cancer types and further explanation on classification of cancer subtypes. This research will investigate the integration of single omics data using selected integration method, and the integrated single omics variation data will be used in the classification of breast cancer using 1D CNN. So here is the introduction to the integration of single omics variations data. The multiple levels of omics approaches can include genome, epigenome, transcriptome, proteome, and metabolome. So the examination of integration of single omics variation information alongside with a clinical data set that has taken the front seat in determining valuable bits of knowledge into the cell capabilities. So the integration of single omics variations data provide a clear understanding on classifying disease according to its subtype. So here is the classification of breast cancer. So the first sign of a tumor is it will form a typical a lump. It caused by the abnormal cell growth. So we have two types of cancer, which is malignant and benign. So here are the few cancer cells: carcinoma, sarcoma, blastoma, and germ cell tumor. So we also have breast cancer diagnosis techniques, which is commonly applied to identify the type of cancer. So one is a biopsy, second is a mammogram technique, third is magnetic resonance imaging, and the fourth is chest X. So next, I would like to explain the data integration technique that we have to use to integrate the single omics variations data. So we have a few techniques which are cluster based, neighborhood based multi-omics clustering, and also similarity network fusion. So cluster base is a method that has been developed to overcome the limitations in the cluster plus. Neighborhood based multi-omics clustering is a straightforward approach which we can use to the integration of single omics variation data that will be built on SNF. So compared to the other integration approaches, NEMO is a simpler than the other current algorithms. So next is similarity network fusions. So it is a network for each data type into a single similarity network. SNF can avoid dealing with multiple scales and also collecting bias and noise in variety of data formats. So next here is the feature selection. So here we have to apply the feature selection because we are using a large amount of data. So feature selection is very important to reduce the data dimensionality and also the complexity. So it will discard the irrelevant and noise data and also which will minimize the complexity of the classification process. So we have a few feature selection techniques that can be applied for the integration of single omics variations such as chi-square test, random forest, depth feature selection, correlation based feature selection, and support vector mission recursive feature elimination, which is SPM-RFE. So here are the previous research on cancer classification. So we have first, which is machine learning technique for breast cancer predictions with 97 accuracy. So the second research is on detection and classification of lung cancer using CNN and Google Net with 98% accuracy, and diagnosis of breast cancer with stacked autoencoder and subspace KNN with 91.24 accuracy. And last one is a survey on gene expression data analysis using deep learning methods for cancer diagnosis with 91.7% accuracy. So next one is the deep learning algorithm. I have listed down 4 deep learning algorithm, which is ANN, DNN, 1D CNN, and RNN. So ANN is a simply neural network, a method that has been created as speculations of numerical models of biological nervous system. So it has 3 main layers, which is hidden layer, output layer, and also the input layer. So we have deep DNN. DNN is a deep neural network. So it's one of the artificial neural network with different hidden layers between the input and also the output layer. So we have 1D CNN. So 1D CNN consists of convolutional layers, pooling layers, and also fully connected layers. And then we have RNN. RNN as a data connection between nodes form a directed graph. So it has the ability to model the progression of a sequence by defining the relationship between current and previous. So next, I would like to explain my chapter 3, which is a research methodology. So this chapter is consists of the research framework, which is represent the entire activities that carry out throughout this research. And this chapter is very important to because it gives an understanding on the flow of the process. So this research framework is consists of 4 key phases, which we will look into next. So this is the research framework. So the first phase is a research planning and initial study. We will identify the topic, we will do the literature review and problem identification, also data collection. The next one is data preparation and pre-processing phase 2. So here we will collect the data and we will pre-processing, which is a data cleaning process. And here, this is the data type we use for this research. We use DNA methylation, miRNA, STNV, and also the clinical data. So that next one is phase 3. So in phase 3, first we will do the data integration by using two different data integration methods, which is concatenated and also the SNF. After we have the integrated single omics variations data, we will proceed to the data aggregation. After data aggregation, we do data normalization and then feature selection approach. After feature selection, we will develop the 1D CNN model. In phase 4, we will do evaluate and analyze the performance of 1D CNN in terms of accuracy. So we will divide the data into two, which is testing data and also the training data. So here is the performance measurement. We use confusion matrix to measure its accuracy, sensitivity, and also precision. So for confusion matrix, there are four important things, which is true positive, true negative, false positive, and also false negative. The software requirements for this research is Windows 10, Google Colab, Microsoft Excel, and MicroOffice. Hardware requirements is Intel Core i5 processor, 32 GB of RAM, and also 5 GB of free disk space. Next, I would like to explain my chapter 4, which is research, design, and implementation. So this chapter involves data transposition, data cleaning, data filtration, data encoding, and data integration and the visualization of data, which is carried out to ensure the results generated are accurate and also not overfitted. So feature selection also applied in this chapter in order to reduce the data dimensionality, and after that we develop one this. So the data preparation data set is took from linked omics website. So here I have attached the website linked, and this research involved of 3 separate omics data set and also 1 clinical data set, which is gene level DNA methylation, miRNA expression, SCNV, and also 1 clinical data set. So the data pre-processing is very important step for the analysis of integration of single omics variations data, because data sets will be highly affected by the negative factors such as noise, missing values, inconsistent extra data. So the raw data set, which is taken from the linked omics, will be processed using Google Colab. So here's the data transposition. Data transposition means we will transpose the column and row. So column will become row, row will become column. So here is the number of samples and number of features for each of the data sets. And then next we apply data cleaning. Data cleaning is a process where we impute the missing values. So here we have a missing values on the DNA methylation. The missing values for this research has been imputed using the standard deviation. Next is data encoding. Data encoding is a process where we change the numerical representation, converted into numerical representation, basically in the binary form. So here we have a multi-class name PAM50 consists of luminant A, luminant B, Bessel, and HER2. So we encode luminant A as 0 and luminant B as 1 by excluding Bessel and also HER2. So the none values that present in the data set was replaced with the most frequent values. So here is the initial counts of clinical data, luminant A, luminant B, Bessel, and HER2. After that, we have a number of samples after we dropped 2 columns, which is Bessel and HER2. And then we encode the luminant A to 0 and luminal B to 1. After that, a data filtering method has been carried out. Data filtering is a very important process because it enhances the data quality by identifying biological relevant features. So it will remove and focus on the relevant data only. So here, after the filtering, data filtering is applied on the 3 omics data set. So before filtering, there are 314 DNA methylation, 324 mRNA, and also 1,080 SENB. After data filtering, we have 167 common patient side. Next is a data integration method. Data integration, where we integrate all the data set, all the omics data set and the clinical data set. So here we apply two types of data integration method, which are concatenated data integration, which is considered as traditional method, and we have similarity network fusion. So concatenated data integration, the concatenated data integration, it involves DNA methylation, mRNA, SCNV, and the clinical data set. So all this data set were integrated to perform analysis of breast cancer. And then this integration was performed using pd.concate function, merging all the data frames alongside the specified axis with an inner join retaining only patients present in all data sets. So each patient's data from different omics and clinical data sets were combined side by side. So here is the number of samples and number of features after the concatenated data integration. Next was the similarity network fusion. So SNF can integrate several data types in the most effective way, making it a suitable choice for this purpose. So it builds a distinct similarity networks for all types of data and uses a complex fusion method to repeatedly merge them. So it combines the single omics data sets, which is SCNV, DNA methylation, and mRNA, also clinical data set. So here is the number of samples and number of features after the integration of single omics variations data. Next, data aggregation. So it is performed in order to combine the information from multiple data points to create a summary of the data set. So here is the feature size and target size for concatenated and DNA and SNF method. So next, data normalization has been carried out. So the data that we are using must be normalized after pre-processing to maintain the range from 0 to 1. So the model could be learned quickly as a result. So data normalization will make the query process easier and reduce the number of data modification errors and get rid of redundant data. So here min-max scalar method has been used to calculate the normalization, and the fit function was used to determine the minimum and maximum values for each data set in order to calculate the normalized value. So here is the result of data normalization on the concatenated data integration before and after. So here is the result of normalization on SNF before normalization and after normalization. So next, feature selection. So actually normalization has been applied after we integrate the data, and then normalization after that feature selection. So as I said previously, feature selection is applied to minimize the dimensionality and improve the predictive model. So it only focuses on the strong correlations or predictive abilities and enhance the effectiveness of the predictive model. So for this research, we use SPM RFE algorithm to achieve our goal. So SPM RFE has been widely used in integration of single omics variation analysis, which combines a recursive feature elimination with SPM. So here is the result before we apply SPM RFE on the concatenated data integration method. So after apply, there were 10,000 data only. So here is the value for of SPM RFE on SNF data integration. So before apply, there were 167. After apply, 150. And then we have to apply synthetic minority oversampling technique to balance the data set, the imbalance integrated data set. So this is the smooth which is applied on the concatenated data integration. This one is before we apply smooth. This one is after applied smooth. And then the smooth is also applied on the SNF data integration. So here is one before smooth and this one is after smooth. So 1D CNN, input layers of 1D CNN. receives the feature selected in the feature selection step as a sequence in 1D array. So an input layer will prepare data for further processing. Convolutional filter will capture local patterns and motifs in the data, allowing the network to learn hierarchical representation. The output of convolutional layer is a set of feature maps representing the learned features at various levels of abstraction. After the convolution operation, an activation function is applied, and then the commonly used activation function in 1D CNN is rectify linear unit, sigmoid, and time. So the activation functions help the network learn complex relationship between input data and target labels. So we have a pooling layer. So the max pooling is commonly used when the maximum value is kept within a small window and the rest are discarded. We have pooling, which reduces the spatial dimensionality of the feature map, improve the computational efficiency. And we have fully connected layer. After that, we have output layer, which will produce the which is last layer of 1D CNN and produce the classification result. So the development of 1D CNN. So the model development divide into two, which is learning phase and also the testing phase. So 1D CNN model implement class using Python programming language. So for this one, we import all the required libraries and we start to develop the model with the input layer followed by multiple convolutional 1D layers by increasing the dilation rates from 1 2, 4, and also 8. So the each curve 1D layers followed by the batch normalization and dropout layers is used to enhance the generalization and prevent the overfitting process. So 1D CNN model is then compiled using Adam optimizer and with the binary cross entropy loss function and also the accuracy as the performance metric. So the random forest classifier is also used and we find the cross validation or an F1 score. So after we develop the model, so the output that we get is as for concatenated data integration, we get 66.67 accuracy, and for the SNF data integration, we get 74.29%. Next is chapter 5, which is about experimental results. So this chapter will discuss about the accuracy, the result obtained from the previous model. So here is the proposed 1D CNN model performance. So for concatenated data integration, actually the training and testing data set is divided into 75-25%. So this is the data division. And we use confusion metrics to extract the accuracy, precision, recall, everything. So this one is considered as true negative, false positive, false negative, true positive. So the true negative indicate the correctly prediction number in luminant A class. The false negative represent the incorrect prediction number of luminant A class, luminant B class, sorry. So the FP, the false positive, is the incorrect prediction number in luminant A class. And true positive is the correct prediction number in luminant B class. So this is the result that I obtained from 1D CNN model using concatenated data integration method. So here is the confusion matrix with the accuracy of 78.79% and sensitivity is 16.67, specificity is 92.59, precision is 33.33 and F1 score is 22.22. And this is the 1D CNN model loss and 1D CNN model accuracy for the concatenated data integration method. So this is the result that obtained for the 1D CNN model performance using SNF integration method. So we obtain an accuracy of 82.86%, sensitivity of 20%, the specificity is 93.33, precision is 33.33, and F1 score is 25%. So here is the model loss and model accuracy for SNF data integration method. So next, I would like to explain chapter 6, which is the conclusion of my FYP. So first of all, I would like to explain my achievement of project objective. So all the three objectives are related to the development of classification model using deep learning methods for address, although the result that we obtained did not meet the expectation fully. So the 1D CNN classifier produced a reasonable classification accuracy for breast cancer based on integrated single omics variations data. But also it produced overfitted graph due to the less optimal 1D CNN's model performance for binary classification. Next, so the more suitable, this method, actually the 1D CNN is more suited to the medical imaging research, suggesting the alternative models might be better for this type of data. 1D CNN architecture contributed to a deeper understanding of deep learning application in the breast cancer field. So single omics dataset was successfully integrated using concatenation and similarity network fusion data integration method to obtain the integrated single omics variations data. So the research constraints are I encounter problems while installing the software like TensorFlow library, and then it also very challenging to study the previous work on 1D CNN for binary number classification since it is mostly applied for image classification data. Next, the future work. So actually we can use advanced, in future we can use advanced data imputation techniques apart from standard deviation, like we can use chain equation, k nearest neighbors, matrix factorization, which can produce a more accurate data. And also, in this research, actually, I conducted binary classification by excluding Basel and HER2. But I suggest for the next, the future research to not exclude the Basel and HER2, like include the multi-class classification, because we should not ignore the rare cases. Actually, Basel and HER2 is the rare cases. That's why I ignored that. So the previous researcher must include the rare cases also. And also we should explore more deep learning methods which can be capable of producing optimal result for binary classification. And we can use cloud assisted virtual machine or graphics processing unit, which can optimize the computational model effectively. So that's all from me. Thank you.